Good morning and good afternoon, everyone. This is Mahalea Marshall from the IQConsortium Secretariat. I'd like to thank you for joining this morning's webinar entitled Predictive Dissolution Models for Surrogate Release, an industry position presented by the Analytical Leadership Group's Dissolution Working Group. IQConsortium was established in 2010 as a technically focused not-for-profit organization and is comprised now of 50 pharmaceutical and biotech companies. Our vision is to be the leading science-based organization advancing innovative solutions in biomedical problems and enabling pharmaceutical companies to bring quality medicines to patients. And our mission is, as a technically focused organization of pharmaceutical and biotech companies, IQ advances science and technology to augment the capability of member companies to bring transformational solutions that benefit patients, regulators, and the broader R&D community. And with that, I'll turn things over to today's moderator, Himanshu.
My name is Himanshu Gadin. I'm from Bristol Myers Squibb. I'm also a member of the IQ Dissolution Working Group. I'm pleased to welcome you all to today's webinar. Today, we are honored to have 6 distinguished scientists from leading pharmaceutical companies. Let me briefly introduce to all our speakers. We have Melanie Dumaret from AstraZeneca, Tessa Carducci from the Merck, Yenmei Elaine from the Takeda Pharmaceutical, Nicole Jabrenko from the Eli Lilly, Anna D. Silva from Johnson & Johnson, and Sarah Nielsen from Johnson & Johnson. I'm truly excited about today's topic. Without further ado, let's begin. And I would like to invite our first presenter, Melanie, to start today's presentation.
Hello, everyone. So as you know, this presentation is meant to summarize the paper we authored from the different industries. From the different companies from pharmaceutical industry, where we try to position how we think about predictive dissolution modeling for release. And if we go to the next slide, you can see that our part of our paper tried to describe a generic approach on how to develop those predictive dissolution approaches. You might have seen already many individual cases where researchers have successfully developed a predictive model for a specific case, but in this paper, it's more about going through the generic steps that you would have to do for every product. As you can see from the flow chart below, the first step is always understanding your manufacturing process very well. Know what changes in your manufacturing processes will affect the dissolution of your product. And secondly, you have to understand your dissolution process thoroughly. What will be the rate limiting step in your dissolution? What's the mechanism behind the dissolution? And whilst you're doing that, you also need a dissolution reference method, like the traditional USB method, developed to make sure you see all the changes you do in your process. And that's kind of a cyclic circle to go back and forward until that's finalized. Once you have thorough understanding of both your dissolution and you have a good dissolution method, you can start selecting the inputs and the outputs for your model and decide which data you want to include in that model. You can generate new data really for that purpose, which is designed to do that, or we can also use historical data given if it's suitable. Then we use that data to calibrate the model, and of course we also have to validate it. During this presentation, all the other speakers will go through different case studies to exemplify this workflow. Once we developed the model, that's not the work done. Then the next important step comes, which is the implementation. And it's myself who will talk a bit about the considerations when we implement these kind of models for real-time release. We have to think about sample size and frequency, how will we use these methods, and how we will capture the data. And another important topic is lifecycle management. It's important that the method remains fit for purpose across its whole lifecycle, and that needs periodical testing, sometimes updates. We need to have contingency procedures. And some of these items will also be highlighted in the case studies. After we went through all the case studies, Jan May will give us an overview of regulatory acceptability for these kind of predictive dissolution models as real-time release test, and a reflection on global harmonization. And then Sarah will end with challenges and the outlook for these approaches, as well as the conclusions. But first, I will give the word to Tessa Carducci, who will talk about the value and drivers for predictive dissolution modeling.
Thanks, Melanie. So as a continuation of the introduction, here's a look at how we traditionally evaluate candidates for surrogate or RTRT. We generally look for dosage forms that are immediate release and have high solubility, as well as rapid or very rapid dissolution. Usually these are belonging to the BCS class 1 or 3. However, predictive dissolution modeling could open the door to a full RTRT strategy for those products that are low solubility in BCS class 2 or 4 and have higher dissolution criticality and complexity, where building a justification for surrogate test replacement is not straightforward. So then here's a look at how the addition of a predictive dissolution model can transform the release of a drug product. On the left is a traditional release scheme. As you can see, there are several quality assurance tests that are performed in the lab after the manufacturing is completed, involving sample dilution and are chromatography based. Moving some of these tests to in process on the shop floor can enable a partial RTRT strategy. Then with the addition of either a surrogate, if that is applicable, or a dissolution model, this enables a full RTRT strategy where all tests are performed on the shop floor. And this is where the benefits, both financial and strategic, are maximized. Then moving to the next slide, we can capture this value of moving from a traditional release platform to rapid release, where almost all of the tests are performed in process on the shop floor, then to total RTRT, including a predictive dissolution model. So we would look at the lab cycle time savings, as well as the cost of API to capture the drug product batch deferment. Then, as you can see on the plot on the left, the time savings increases as more and more tests are replaced with surrogates or models. Then on the right, considering the batch forecasts for a product, the FTE savings over the life cycle of the product can be estimated. More importantly, there are several strategic benefits of utilizing a predictive dissolution model, including deeper mechanistic understanding, automation, data integrity, and safety benefits. As you'll see from the case studies in this webinar, you gain a deep knowledge of the dissolution as well as the sensitivities throughout the model development process that is sure to pay off, ensuring smooth and high quality supply of a product. We'll now move into those case studies, and so I will hand that back over to Melanie.
Yes, the first case study is the most straightforward one. We had a drug substance at AstraZeneca, which had very high solubility and permeability. So it was a BCS1-like drug substance. And of course, we first started with a lot of process understanding on our product, first starting with a risk assessment. Then we changed the components in the formulation to understand the effect of the different excipients. And then we also varied parameters of the process to test robustness. In parallel, my colleagues from Biopharm were developing dissolution understanding, and they understood that this was an immediate dissolution and that actually the rate limiting step was disintegration. And they also discovered that disintegration was more discriminatory than dissolution. So for this molecule, we would have had 3 options on how we could use. We had 3 different release tests we could have used. As all the API dissolves immediately, the dissolution value is the same as the assay value. So we could have had a model based on the drug substance content by NIR in the powder blend and tablet weight. We could also use an at-line disintegration method, which is also very fast and can be real-time release. But now I had very innovative colleagues from the engineering department who developed a model based on tablet porosity and coating thickness to predict disintegration, and that was considered the easiest approach. And now we will go to Anna for a bit more complicated study.
Thank you, Melanie. Good morning, good afternoon. So I bring you the case of a case study number 2 from Johnson & Johnson. And at Johnson & Johnson, we first can try 2 different approaches for our predictive dissolution models. So first we have the chemometric approach, in which we have a PET tool, which is an NIR instrument that collects spectra from the tablets. And then those spectra together with CMAs, CPPs, and CQAs, if they are needed, they are input into a PLS model in order to predict our dissolution for that tablet. On another hand, we can also develop what we call a conditional approach, where those NIR spectra of the tablets are not used directly, but they are put into a PLS model to predict drug substance content, and then that drug substance content together with different CMA, CPP, CQAs that depend on the product. In this case, it was drug substance, PSD, and hardness that were driving our dissolution. And then they are linked via a conditional model to our dissolution. So how do we calibrate? So we select different batches with varying CMA CPPs to intentionally create different dissolution rates, depending on what is discriminative to that method, dissolution method. And then we take spectra of 6 tablets, NIR spectra, and then we, on those tablets, perform traditional dissolution testing. And then we can establish our PLS or conditional models to establish the relationship between the inputs and the outputs. I already talked in the previous slide about the inputs. What is the output? Dissolution. But how does that look like? It can be either that we predict individual dissolution results for each time point independently, or we can instead predict the parameters of a mathematical function or first principles model that actually describe our dissolution curve. What's different between these 2? I think at some point people have heard of PLS, of partial least squares model, which is readily available. So that's rather simple to get a hands-on. A conditional approach is more complex, requires more statistical expertise. But on the other hand, your model coefficients can be clearly interpreted as the effects of each parameter that you take into the model into the dissolution. The chemometric approach has the complexity that it might require additional robustness measurements to make sure that not only the parameters that influence the dissolution, but also other parameters that can influence the NIR, but not the dissolution, are covered for, and that they are not influencing the performance of the model. This is not needed for the conditional approach. Yeah. Then we have our model. How did we validate it? So for validation, instead of n equals 6, we used n equals 12. And we had 6 additional batches where we again varied the relevant CMA CPPs in order to create different dissolution rates, and then also a batch of placebo. And then we validated according to the ICHQ2. And for the chemometric approach, we actually validate all parameters, so accuracy, linearity, precision, etc. I will go more in depth in the next slides about this. Or for the conditional approach, there is a difference that we believe we don't need to validate for precision, specificity, and robustness anymore, because as I mentioned, it is the first step is a PLS model for the prediction of drug substance. And since for that model, we already validate these parameters, they are dependent on that first model. So we don't need to revalidate for this. Then I will go in depth into each of the validation parameters. So for accuracy, as I mentioned, we do n equals 12, n equals 12, sorry. And we evaluated based on a comparison between the prediction of the model and the traditional dissolution for that same tablet. And then we can calculate metrics that are pretty standard, like the standard error of the prediction, or others such as the bias of the bias corrected standard error of predictions. Linearity, we use exactly the same set of tablets, but then we draw the calibration curve, we visually inspect it to see is it linear, is it not. We report both the Pearson correlation coefficient, and we can also report the determination coefficient. The range, it's simply also on the same set, but it's simply the upper and lower dissolution results as determined by the reference method. Specificity. We take the spectra that we collected from the different tablets, we make some overlays of the spectra versus a spectra of the pure DS and a spectrum of a placebo tablet. We also plot the regression coefficients and the loadings for the model. And then additionally, also the spectra collected from tablets that contain the active, they need to fulfill the thresholds for spectral quality attributes like Malanovis distance and residuals. And for the placebo, of course, this cannot meet these criteria. Precision, we do precision at 3 levels. So we can do system repeatability, in which we place one tablet on the instrument, measure it 10 times. We can do analysis repeatability, where we place the tablet again 10 times, but in between we actually remove it from the instrument. And we do as well intermediate precision, in which we repeat this analysis repeatability in a second day. And then we go and we evaluate the pool standard deviation, absolute difference, or we can also evaluate the pool relative standard deviation to get criteria for these parameters. Robustness. Robustness is actually performed during method development. So as I mentioned before, we need to make sure that parameters that don't influence the dissolution are not influencing your dissolution output. And therefore, we test the robustness during our method development. A way that we evaluate the robustness is by looking, of course, to the accuracy of the predicted results, but we also use metrics for this, such as the Malanovis distance and residuals, which I've mentioned before, to identify potential outliers. How do we actually set criteria numbers for all of these parameters? So how we set it is that our analytical profile for this type of models is actually that we aim that at the specification time point that is selected, the predicted dissolution at that time point needs to be within a 5% from the true dissolution. And true dissolution, we never know what the true dissolution is, but we will take it as being the dissolution by the reference method. This, of course, for time points for at which 50% of the API has been dissolved. And where do we base this? Where does this 5% come from? So it actually comes from the standard reproducibility criteria that we apply for a traditional dissolution method. Okay, so I give it to Nick von Zabareco to present this case study from Eli Lilly.
Thank you, Anna. As Anna said, my name is Nick Zabarenko, and I'll be talking about a case study that we worked on at Eli Lilly for developing a PDM for real-time release of a batch process using offline and at-line inputs into the predictive model. So the process in question involved a drug substance with BCS class 2-like properties, which means poorly soluble, although with good permeability. But it was enabled through an amorphous spray dry dispersion So there was an intermediate stage before tableting. And as you can imagine, the spatial dispersion's particle size has very little correlation to dissolution because across the range of the particle size manufactured for an SDD, it was all fine particulates that are non-spherical, non-solid particles, no correlation between that and dissolution. The process was batch manufacturing, aiming at 2 commercial dosage strengths, and was following an accelerated commercialization plan. So no white space in the Gantt chart, very rapid plan to go from early trials and development through to launch. But we wanted to minimize the amount of testing burden. So there was a comprehensive RTRT plan, including multiple CQAs, assay, identity, and dissolution that were all explored throughout the development process. So, of course, where do we start? We start with what parameters are actually needed to feed into the models, meaning which material attributes, which process parameters are critical in a sense that they can influence the result of a CQA. And the flowchart says CPP or non-CPP. We lumped CMA into the same terminology. Essentially, all of the variables in the process were evaluated using this flowchart. So by looking at the previous experience, at understanding scientifically the different unit operations, and at early small-scale development tests, we could group the parameters, or variables rather, into low risk and not low risk, medium or high. The low risk ones essentially was just multivariate testing to confirm that they would have no effect and thus not a CPP towards the CQAs that the models would be used for. For the other parameters, it was a little bit more of a complicated decision tree to determine whether they had an effect, and if so, whether it was a big enough effect that could lead to CQA failure. And we then used a combination of univariate tests for individual high-risk variables, as well as some multivariate and design of experiments studies to determine which variables were CPPs or CMAs, in this case specifically for the dissolution process. So with that being said, what you are seeing here are dissolution profiles of various core tablets manufactured or produced during development with various parameters and variables changed. And what we saw was that there were essentially three areas of criticality. One was on the intermediate, the spray-dried dispersion intermediate side, which was the crystallinity. Any amount of crystalline material was essentially insoluble in this dissolution method. And the dissolution method was developed to be discriminating for what we consider to be clinically relevant variables. So crystallinity meant the drug is not available to dissolve and to release. You need the content to be fully amorphous to get the full drug out. And then that is the essentially y-axis scaling of your dissolution profile. The shape of the profile in the x-axis scaling was fully determinable by tablet solid fraction, but it in itself was dependent on both granule solid fraction and tablet compression strength. So those profiles are very well described by a mathematical Weibull function. So it's a non-mechanistic function, but the parameters can be regressed to process variables or material attributes. So A is just the scaling factor on the y-axis. As I described in the previous slide, crystallinity was pretty much the main crystallinity was one of the contributors to it. The other one is the tablet label claim. If you have less drug in there or more drug, obviously it'll scale for your total amount of release. The shape of the curve then is described by 2 parameters mathematically, and those parameters were fitted with only the critical parameters of compression stress for the tablet and the ribbon solid fraction of the roller compaction granulator. So those 2 processes describe the entirety of the curve. plus the crystallinity and label claim describing the entirety of the dissolution profile for core tablets. So how feasible is it to have a real-time release, or at least a predictive model? Well, as I mentioned earlier, we were working on real-time release strategy with multiple CQAs, one of which was assay. And near-IR spectrometry was being developed as the model, as the spectroscopy tool for modeling the total label claim within the tablet. And it was a well-developed model, was able to predict the label claim with very high accuracy and reproducibility. The more challenging part was the crystallinity, which did not factor into the assay model, but does factor into dissolution. But one of the regression vectors, I believe was a second derivative on near-IR spectroscopy, did show that on crystal material there were peaks observed that were not found in the amorphous material or in the placebo matrix. So thus, those peaks were then used to develop a quantitative analysis of crystallinity, and a single near-IR spectroscopy measurement on an individual tablet could then be used to predict the parameter A for that tablet. Combined with the batch scale parameterization of lambda and kappa, we then had the full prediction of dissolution profiles of core tablets. Note I keep saying core tablets, because once we started coding those tablets, we noticed the dissolution would change. It would change for the better in the sense of being more rapid to release, but now it shifts away from our model. And interestingly, we saw the same performance change upon the core tablets stressing on stability. So at increased temperature and humidity, there was an annealing process happening within the tablets that would then improve the release rate. So how do we capture that? Well, more batches, more tests to then feed into a statistical evaluation. And we found that an adjustment parameter, a delta, on the lambda and the kappa parameters could be found such that by using the coding parameters of exhaust temperature and relative humidity, as well as the initial parameters defining the core tablet profile shape, the deltas on those parameters could be very well modeled. So with this combination of near-IR spectroscopy, coding, tableting, and roller compaction parameters, we had a fully described dissolution model for coded tablets across our entire design space. So this is what the overall chart for the dissolution model looks like for us, with the coded tablet dissolution being calculated from this Weibull function, which itself is fed by a tablet spectroscopy model, or tablet spectroscopy, a PLS model predicting the A parameter, and then a batch level process values feeding into the kappa and lambda and their deltas from the coding. We used this model, we built it incrementally from early development batches as we went through the multiple scales of this development process and into the clinical testing. And as we got to the clinical manufacture, we actually used this model to incrementally predict the parameters of the next unit operation, meaning that granule solid fraction coming out of the roller compactor was then used to inform the tablet compression force ranges that we knew would give us the desired core tablet dissolution profiles, which the compression forces then were used to predict what the coding range of humidity and temperature had to be to give us acceptable CQAs on the released final tablets. And so the same process was used for the first lot at the commercial scale and the process validation batches. And this led to no technical deviations, zero investigations from the point of view of dissolution, flawless execution of the scale up and transition from development through clinical to commercial scale. However, we did not use this model for real-time release, so we did not submit it into the NDA due to both internal and regulatory hurdles. So the amount of data and the type of data required to feed into this model required an informatic infrastructure that was not available at our manufacturing site. And at the pace we were going to meet the launch timeline, we didn't feel it warranted the investment to put in that infrastructure. Additionally, it was perceived that model maintenance would be a greater burden than method or process maintenance due to the need to measure intermediate materials such as ribbons that were not necessary during regular design space maintenance. Finally, we got feedback from regulatory agencies that they did not consider the model to be in vivo relevant because it predicts the level of crystalline material, but there is no direct link to the level of bioequivalence because we didn't take different crystalline material into the clinic. We simulated in vitro what would happen. We simulated in silico, but we didn't make any batches for bioequivalence or relative bioavailability with crystalline material. And so batches representing the extremes of dissolution in the in vivo safe space weren't tested in vivo. And this was acceptable for the dissolution method, but not acceptable for the model from the regulator's point of view. So additionally, the expectations that they have voiced for model calibration and validation, the number of batches at scale were high burden for implementing this model in the time required for launch, and the amount of work for a post-launch change request would have been quite burdensome at the time that the project was being considered for NDA. Additionally, with the multiple agencies and divisions of agencies around the globe, there were different levels of feedback we got, and the need to modify the model at those different points was just perceived as too high of a burden for it to be useful. But it is being used internally as a check on our process and was very helpful during the development and scale-up. So with that, I'll turn it back over to Melanie for the implementation of real-time release models.
Yes, so now we heard all about how to make the model, and now I'm going to talk how we're going to actually use it. The examples from Anna and Nick were using tablets, and that's the first thing we can take into consideration is the sampling. If we have tablets, we heard that they took 12 tablets, which seems to be the standard for these real-time release tests, but I never found a strong justification why we need 12 tablets for real-time release test. But on the other hand, it's so fast to do the measurements, so probably it's no extra effort and you get more information on your data. So for tablets, that's the most approach that's used, and those tablets are then measured at line, which is still very fast. That's why it allows real-time release testing. If we go to continuous direct compression, for instance, we can base the real-time release test on in-process measurements. I think it can be done for batch as well, but maybe a bit more challenging. And if we look at the type of measurements that are typically used, it's the NIR spectra of powder blend, tablet weight, process parameters during the process, and wet granulation, also torque. And mostly we have these parameters at very different frequencies. Like an NIR spectra we have every 1 second, while the tablet weight only 15 minutes. So then you have to do a smart averaging or correlation between those two data on how to connect it and get a representative measure for tablets in your batch. Then, no, can I go back, please? Then another challenge is that instead of 6 or 12 tablets, we might have thousands of predicted dissolution values. The question is, do we average across the entire batch, or mostly that's not acceptable by regulators, but they propose some things like process windows where you split the batch into different time windows. And it's also good to consider that all the testing is on the manufacturing floor. And if we can go to the next slide, because that's where the next implementation challenge comes. So now it's not the analyst doing the analysis, but it's the operator on the floor that also has to do some process measurements. So they have to be trained for that. An ongoing discussion in my company often is who will own those instruments on the manufacturing floor. Also, the operators need to be very well trained on how to handle the information they get during the batch. Or you can also automate some of the things. If you get an outlier in your NIR measurement, then you don't want to fail your real-time release test, but you will have to follow another procedure. Although if you get an outlier in the dissolution predictions, that's probably a real deviation that will have to be raised. The operator also has to know, what do I do if my in-process instrument is not available? Is there a backup method? And if we automate the calculation of dissolution, things we have to consider is to not include the diverted tablets in those calculations, because they don't end up in the eventual batch, so are not representative. And another important part of the pharmaceutical quality system on the site is the lifecycle management. As I mentioned before, the RTRT, the real-time release test, has to be fit for purpose along its entire life cycle, and we achieve that by doing periodic verifications with the reference methods. We also should have very well-defined triggers for model updates, and it's also good to have good procedures for post-approval changes if needed. If we go to the next slide. Nick also highlighted this as a blocker in his application, but as you heard from the examples, we process a high amount of data, and there has to happen a lot. If you can do it all manual, like the top row in this figure, where you would do the data capture and storage in the instruments and some basic pre-processing, and then you can take it out and do it in your separate software, a calculation to calculate the dissolution value, and you manually transcribe it in a report. Of course, that's a lot of risk for human error. You could automate those scripts, which reduces the risk further and makes it more efficient. Many companies now have a PAT management system or a data analytics platform in which this whole data processing steps and capture are automated, and it can be automatically sent to the LIMS system. Of course, that's a higher cost than to implement, but it does decrease the risk of transcription error. And I think that was it for me, and we can now go to Jan for the regulatory acceptability.
Sure. Thank you, Melanie. Hello, everyone. This is Jan Milan. Our next topic is a quick overview of regulatory acceptability and global harmonization. In general, regulatory expectation for dissolution testing of immediate release drug products are in large part based on their pharmaceutical classification system class. As we all know that discriminatory power and clinical relevance are the main aspect of the dissolution method. The method parameters must be justifiable, and it should be robust and follow global regulatory requirements. These apply to RTRT method as well. The use of enhanced product and process understanding in combination with quality risk management to establish an appropriate control and release strategy. These include surrogate for release testing for various critical quality attributes. This is supported by health authorities. The desire to speed delivery of pharmaceutical products to the patient globally incentivize the use of modeling in layers of traditional analytical laboratory testing for QC and drug product release. So predictive dissolution modeling, PDM, has been emerging recently as a novel surrogate for the dissolution CQA. Development of a PDM requires a thorough understanding of the release of the drug substance from the drug product, as you have heard from the cases examples. One thing we do notice that the use of PDMs, a surrogate for dissolution drug product release, is less common than the models of other critical quality attributes for drug product. And in the ICH Q14, the new ICH guideline, real-time release testing is considered a type of analytical procedure, so which allows for the assessment of the quality of the drug product based on process data and in-process controls. This enabled the release of the product without a need for post-manufacturing testing. And this approach will enhance its efficiency and ensure that the final product meets quality specification based on continuous monitoring. Let's next slide, please. The use of PDMs as surrogates, as mentioned, for dissolution release is less common than real-time release of other drug product critical quality attributes due to various challenges summarized in this slide. First is the scientific and technical challenges, such as availability of predictive surrogates. And the development of a dissolution control strategy for a drug product has so far been highly product-specific. Routine workflow is difficult to develop. Also, it's challenging to transfer a model calibrated using drug product manufactured at smaller scale and then to commercial scale. And also lack of regulatory guidance on model validation, unclear global market acceptability, and uncertainty in the value of developing and implementing the PDMs. And additionally, low solubility drug products are poor candidate for dissolution surrogates because of increased regulatory scrutiny. But you have heard the story example from Nick with the PCS class 2 example. This can be done. Small variations in drug product attributes may have potentially large impact on bioavailability that would not be adequately captured by the dissolution model. And lastly, potential additional regulatory hurdles to approval will impact the business decision of a pharmaceutical company to develop RTRT. Next one, please. Some recommendation consideration to improve global implementation and acceptance of PDM for real-time release testing. During method development, we should consider the new ICH guideline Q14, the recommendation for method development and method life cycles. Select the right experiment supporting development activities and verified suitability of the sampling and validation plan. One of the very important experience is that engage with regulators across the entire PDM development journey. The sooner, the better. The PDM submission packages should provide a thorough justification of the input parameters and selected model type and parameters. The sampling plan should be statistically justified to ensure that the selected samples are representative to the entire batch, such that potential inter-batch variability can be detected. And lastly, validation results should demonstrate that the PDM-based release is fit for purpose and ensure that non-compliant tablets can be detected. Next one, please. So overall, there are, as mentioned, there are not much guidance, global guidance from regulatory for RTRT. However, there are multiple common global guidelines related to method development dissolution can still be used and to follow when we develop the method for RT release. Yeah, this is just a quick information only. That's all for my presentation. Next, let's go to Sarah, please.
Thank you, Yan-Mei. Yeah, I'm Sarah Nielsen from Johnson & Johnson, and I'm going to present some challenges and outlook moving forward based on our experience with real-time release and dissolution. Thanks, Nick. So I just wanted to kind of bring us home at Johnson & Johnson. This is sort of our typical real-time release strategy. You can see here, we convert as many tests from liquid chromatography to near IR testing or surrogate model. And then for things like peroxide and degradation products, we convert that via statistical analysis to sunset testing via a very specific statistical plan, testing a certain number of batches over a certain number of years. So for us, we've had good success. We've had 2 approved surrogate dissolution models on 2 different lifecycle management products. And what we found was with this strategy, we've enabled a 1 to 2 day product release as opposed to over 30 days with our more traditional methods. So if you could go to the next slide. So with the real-time release in general, we found a lot of strengths and a lot of challenges. And I'll kind of hone in on the dissolution challenges here on the next slide. But for right now, we found the strengths and why we really have doubled down and we're going for real-time release on as many products as possible is that we really appreciate the real-time knowledge of product quality that comes with real-time release testing. So we're either testing our tablets or our blends real-time or outline. So we have insight into our manufacturing product and our process in real time. We found a general acceptance of regulatory acceptance and quality acceptance for tests, assay, content uniformity, and identification. We've had good and successful approval by health authorities here. We have had past challenges, and based on these challenges, we've kind of developed a robust and reliable overall process for getting to real-time release. As I mentioned earlier, we've have a significant decrease in lead time. So we've been able to decrease significantly the time from the end of manufacture to product release using real-time release. We have found some challenges in this. As I mentioned, we've had 2 approvals, but those were both on lifecycle management products. So we weren't really tied to the development timeline. So I'll talk about the challenges of when we've going for real-time release when it's tied to a development timeline as opposed to a product that's already approved and being manufactured. We have found there's some really rigid model requirements kind of coming from regulatory for content uniformity and blend uniformity models, including lifecycle management of those models we found to be a challenge. I think a couple speakers mentioned before me this data infrastructure and automation challenges. So with our approved methods, we were able to create an end-to-end fully automated digital solution. But that's a really challenging project to take on, including, product release. It's the highest level of qualification of a system. You have to tie into other qualified systems like LIMS. So for us, having this digital solution was a challenge. We were able to overcome it, but I could see it being a challenge for a lot of companies moving forward. And as I mentioned just a few minutes ago, the surrogate dissolution model development, when it's tied to a new product, there's really tricky timing here with regard to the model development and the CMC process development. And what I'm trying to say here is that when we kind of finalize our method that we're going to model, that kind of comes late in the development life cycle of a product. So then being able to develop a surrogate model in collaboration with our in vitro model, we found the timing to be sort of tricky and a challenge for us. Also, kind of alluding to the previous speaker, there's a lot of regulatory challenges that we find with surrogate dissolution. We find that the bar can be higher for surrogate dissolution, method validation, et cetera, with health authorities. So we found those to be some challenge. And then finally, the post-launch real-time release activity. So if we want to do a model development and validation, post-regulatory approval of the product, kind of getting that global regulatory approval can be quite a long timeline. So you can go to the next slide, please, Nick. All right. So we're going to now kind of dive into the what I discussed before with regard to the tricky timing. So, we're doing our process development, analytical development, and filing. And in the meanwhile, we're trying to do our dissolution model, design of experiments, our in vitro method development, and then our model development. And sometimes here, the timing just doesn't match up. We'll do some changes to the in vitro method sort of late in the product development, and then that leaves us short on time to kind of redevelop and validate our surrogate dissolution model. So for us, I would say for new products, we don't have a successful filing as of yet because we're still trying to master kind of how to figure out this tricky timing that I've talked about here. So that's the end of my talk. We can go into conclusions now. So in conclusion, we're able to show, dissolution methods, they're often not univariate. It's a multivariate space, which requires knowledge of your process and product and in vitro method in order to achieve a high-performing surrogate model for dissolution. Collectively, we found that the establishment of appropriate controls are essential to ensure that our dissolution surrogate models results are reflective of our product performance and that we're really catching all that we need to catch in terms of our product. We've also found that an understanding of the method, all the equipment you're using, the materials, how to measure for different measurements that kind of feed into the dissolution model, people, environment, there's a lot of kind of mastery of understanding every aspect of your process in order to achieve, like, once again, the surrogate dissolution. And then also minimizing the variability in their operation, we can kind of hone in on a better, higher performing surrogate dissolution model. And then also this will help avoid investigations and redevelopment of your model later in the product life cycle. So with that, we'd like to go to acknowledgements. So the presenters here represent a very large group of people from all of our respective companies, really working hard on these overall strategies to get to that surrogate dissolution ideal state. Anything else? Any of the speakers would like to add? Nope. All right. We can go to questions then.
All right, thank you all. Himanshu, would you like to start the Q&A session?
Yeah, for sure. Yeah. So let me start with the questions came out through via Q&A from the audience. So first question came out like, isn't BCS for case study 1, isn't BCS class 1 module with high solubility dissolution will not be rate limiting at all?
Yes, I can answer that. That's correct. The dissolution is not rate limiting. So I hope I said correctly during the talk that in this case, this integration was the rate limiting step because it's dissolving immediately and we have to wait until the tablet is disintegrated until everything is in the solution. But having said that, that was also very fast. And that's why we also fulfilled the requirements of ICHQ 6A that states we can use disintegration testing instead of dissolution testing. But I wanted to give a bit of context. I got the question before, why did you even start making that dissolution model if you know you can do disintegration? It's again about those timelines. We didn't know from the very beginning. You build that understanding as you go in the process. So by doing a DOE to develop the dissolution model, we actually found out that it's very robust dissolution, and the only thing we ever changed is disintegration. So that was a learning we got during the development of the dissolution model.
So to follow up on that, Melanie, since we can use disintegration as a surrogate, could we use RTRT as a model of disintegration and remove a destructive test entirely?
Yes, that's what we're aiming to do with, so my engineers have a model based on the tablet porosity and the coating, because that were the two factors influencing how fast the tablets disintegrates, and it's ongoing, so we hope it will get accepted.
I will go with next question. So for case study 2, some of the variants might not be biorelevant. Would this renders the model not accurate in predicting the batch quality, such as rejecting a batch which might be B bioequivalent?
Bioequivalent.
Yeah.
So I'm no expert on the solution, but let me read the question again. Some of the variants. Do you mean the parameters that actually your model needs to capture in order to predict dissolution? Because then I would say, if your dissolution itself is picking something which is not biorelevant, yeah, that's a problem of your reference dissolution methods. If you mean the other parameters that we add to the model as robustness for our chemometrical models, the reason that we include them is actually for this specific reason, because if we don't add them, then the model will possibly link them to the prediction of the dissolution, and you might get an inaccurate result. So by putting them in your PLS model, you are making sure that the PLS doesn't see it, meaning it will not influence your result. And it doesn't matter if it's not relevant for your dissolution. It's making your model, your chemometric model, robust to it.
Yeah, and I'll also chime in on that. From a dissolution perspective, we often design the dissolution method and a specification and consider batches that specifically are made to be not in vivo relevant, take parameters that are way outside, just to see what the dissolution method discriminates for. Sometimes it discriminates for things that are process relevant or manufacturing relevant, but not in vivo relevant. And it's all a matter of designing an appropriate dissolution method. So all of the case studies we presented and all of our approaches hinge on having a dissolution method already developed or being developed in tandem, such that the dissolution method follows the guidelines and regulatory expectations, and then the dissolution model predicts what that method would measure. So yes, you would certainly have the risk of rejecting a non-bioequivalent batch. And generally, the regulators will tell you they would rather see you reject a batch that might still be BE rather than pass a batch that might not be BE. They are fine with you being overly discriminating and having some false positives as long as you don't miss any negatives. So yeah, you could end up with an overly discriminating method or overly discriminating model, and that's a risk for it's an economics risk, and that's just something you have to work through the development to see what you're discriminating for, how much of a risk it is when you go to your design space, and then be able to convince the regulators that what you're discriminating for is more tight than what could affect the patient.
Okay, so
And if anyone wants to chime in on that or have other perspectives, so we have plenty of time for discussion, I think.
I can just add that I think if we do have an understanding of a clinical failure point, both the dissolution method as well as the model would be expected to detect that. And like, because they're inherently linked. And I also want to mention that I don't feel that setting a specification based on clinical relevance, like that approach or the predictive dissolution modeling approach, are mutually exclusive. In fact, they can be used together. And right, for a clinical understanding of the dissolution failure point, right.
There's a comment online that from a company perspective, you would not want to be overly discriminatory. And I would agree that we wouldn't, but what defines overly discriminatory varies project to project and varies between industry and regulators. So for each project, you have to decide what is overly discriminating, and you have to work in tandem with regulators and have communications open before you submit to make sure that your definition of overly discriminatory matches the expectations before you put in the model. But you're right, you wouldn't want to be overly discriminatory. But you have to demonstrate that you can discriminate for anything that they might expect could affect patients negatively. So it's a tightrope balance a lot of the time, which is why the success rate of actual real-time release testing of dissolution is so low so far. One of the reasons. Shall we take the next question?
Sure. So next question is also for case study 2. So it says the validation parameter being discussed are for the quality of the spectral information, but how have you accounted for the analytical method quality of the final dissolution prediction? As for example, its accuracy, precision, etc.
So actually during validation and all these parameters, we calculate those metrics I talked about on the percentage dissolved. So the final output is the percentage dissolved for a certain time point, and is on that analytical result that we calculate all of the metrics, not on spectral quality, really on the final result.
And the next question is also for you. How much % variation done from target to CPP to get different dissolution profile?
Yeah, so actually depends on the parameter. So in this case, we had 3 parameters for the case study 2. And for example, the percentage of the content was varied between 70, 130, because our specification, our potential specification, was to have at least 80% at X minutes. So then we make sure to cover that and even broader. Also because we can leverage from the model, one of the options, the conditional model leverages from the already existing model for the prediction of the drug substance content. And those are standardly also developed between 70 and 130% of API. Another discriminating factor in this case was the hardness. And to select a range, it was based on both the norms, but also what is discriminative for the dissolution model, because of course we need to create some variation to be able to model it. So we took these two into account. And for the particle size, actually, of the API of the drug substance is exactly the same. So we took into consideration what is the spec for the particle size for the API, but also the discriminative power of the dissolution method towards the particle size. So it's highly dependent on what your dissolution reference dissolution model is capturing as the discriminative properties.
Thank you. So our next question is for case study 3. From my understanding, PLS is mainly a linear method, while the relationship between NIR spectra and dissolution profile is usually nonlinear. Do you think PLS can still be used effectively in this case? And how would the performance differ if we applied ANN instead, in terms of data requirement, what would be the minimum number of data sets or sample needs? Sample needs, sample needs to properly train a PLS model so that it can reliably predict dissolution profile from an NIR spectra.
So in our case, we didn't actually use near-IR to predict the dissolution profile. We used it to predict the scaling, the Y scaling factor of the dissolution profile, which means we use it to predict the maximum final value. So the near-IR spectrum was used to predict the maximal extent of the solution, not the profile itself. Now, could near-IR be used for fully describing the solution profile? In some cases, yes. You're right, PLS model wouldn't be able to capture that. I've seen work in ANN, in neural network, and lately more AI ML approaches. I couldn't answer as far as how many data sets it would take to train that, because it really depends on what the attributes of the tablet are that actually are driving the changes in the solution and the changes in its spectroscopic response. So the more distinct those domains are, the fewer data sets you would need, but you also need to make sure you've demonstrated that every process parameter and every material attribute that could contribute to either the release or changes in near-IR spectrum are captured in that model. So the answer is as many as you have variables that could influence either the CQA or the input. But in our case, the PLS model was fully sufficient because it was a linear response. It was how much crystal material and how much total material of the drug substance is in that final tablet. That was all that near-IR was telling us.
I would wanted to add something about neural networks as well. I think it will typically require more data because it's more prone to overfitting. So I think you will need a bit more data if you use neural networks. And I think another challenge is that it's more of a black box method, although that's changing nowadays. But regulators really want to know how much every parameter contributes to our dissolution. And I think that might be actually challenging with neural networks to have that very scientific relationship between your input parameters and your finally predicted dissolution. That's a bit my reflection on that. Not to say it's impossible to use neural networks, but I think it would create extra challenges.
Yeah, if I might add my 2 cents as well. So as I mentioned, we try 2 different in our case study, 2 different modeling approaches, in which one is indeed the PLS model directly from the spectrum. And actually, after trying the 2 approaches, both methods had very similar predictive power. Both were suitable for the purpose. So we needed to make a choice. So it means that actually predicting directly from the spectra for the parameters that were the drug substance content, the hardness, and the particle size, it works quite well. So that means from the NIR spectra on the tablets, we could extract the variability related to hardness, the drug substance content, which is no surprise. What was trickier in the end between the 2 methods was to show the particle size, because on the NIR spectra, we could extract information, but it was not so straightforward to show that it was sufficiently captured. For that reason, we ended up going more to the conditional way, because in the conditional model, as I mentioned, all the coefficients of the model, they show exactly how much that certain parameter has an influence. So in that perspective, it was better to go with the conditional. But we still could have a model with the exact same performance with the direct prediction from the spectra, just more in regards to regulatory acceptance and showing that our methods had the same discriminative power as your dissolution method. We ended up going more in the direction of the conditional.
So I will go with the next question. It seems that a question for all panelists, it seems that a question to consider for PDM is Are you developing a substitute for the traditional dissolution test, or are you developing an entirely different method that is predictive of drug product release? Setting specification for the first option straightforward, you could use USP criteria based on sample of tablets, but for the latter, you could feasibly estimate the release of all those units, and there you would need an entirely different approach to determine conformance and acceptance of those units. Any comments on this?
So in general, we don't develop an entirely different method predictive of release, i.e. we're not saying this is a different way of predicting whether something's bioequivalent or not. We are piggybacking on a developed dissolution method. And so all of this is a substitute for the traditional dissolution test, predicting the same value that that dissolution test should measure. But as far as the USP 711 criteria, that is something that we regularly discuss with the regulatory agencies as far as what is acceptable, what is insufficient, and what is overfitted. Because there are regulators that I've talked with who say every tablet that you can take a measurement on, or that you are taking a measurement on, should be used to calculate dissolution because you can. You have the data, you should use it, and you should predict disso for everyone. And then there have been others who've said the exact opposite thing, saying if you have too many samples, you are over-testing a batch, and now you have a different statistical rigor to be able to say whether the spec is correctly set. So that's still an issue that's ongoing. But our position in general is we agree with USP 711 criteria applicable to dissolution test, should also be applicable to dissolution predictions by using the same number of tablets, measuring whatever inputs from them go into the model, and predicting that number of dissolution values.
I would agree with that, Nick, but I think a challenge not mentioned in this question is that for tablets, it's rather straightforward, but if you're measuring on powder blends or on process parameters, I think that's where it gets even more complicated.
And I think we are running out of time.
Yeah.
There were questions about whether these case studies are published. I think all of these case studies were in the publication that was the pre-read included with the webinar description, at least the high level. I can say that the one from my group will be coming out in more detail for the entire process in probably within a month or two. It's currently in review.
Thank you all. Unfortunately, we are out of time, but I'd like to thank our speakers today for sharing their perspectives on dissolution models and their insightful findings through case studies. I hope to see our attendees back at a future IQ webinar. And until then, thank you again to everyone. Take care. We'll see you soon.